Colony collapse disorder is known as CCD, is a serious matter, and people around the world are affected by it in growing proportions. It has been reported that last year in the US alone, over 50% of the beehives collapsed. This discussion today will shed light on beehive complexities and the possible approaches to solving this most perplexing agriculture and human health issue. Here with us from Japan is Ken Hashimoto. Ken is the Director of Research for Yamada Bee Company, the largest producer of royal jelly in the world. Tom Glenn is an expert in bee health and queen bee rearing and comes to us from Fallbrook, California. Bob Danka is a research entomologist at the USDA Research Lab in Baton Rouge, Louisiana. And Danielle Danny is the Hawaii Department of Agriculture's apiary specialist. Paul Apau is with us from Maui Queen Bee Company. I am Lloyd Fischel, and I operate Fragrant Orchids of Maui. Danielle, maybe as head of apiary here in Hawaii State, you can give some kind of a definition of what that term is referred to in the past.
Sure. We have not verified any cases of CCD in Hawaii, but on the mainland, the classic definition of CCD was symptomatic. So that means you couldn't go in and find a pathogen or a problem that killed the colony. The colony when it collapsed, it would be a handful of young bees with the queen, and normally when a colony dies that way, you would see a big pile of dead bees. And here, in a colony that's collapsed with CCD, there's no bees dead in the hive. They've all gone out and disappeared. Another indication is that this hive is full with brood, so clearly it was a booming metropolis, and now there's not enough bees left to support that amount of brood. And a honeybee hive is such a rich resource that when it collapses, there are many marauders that would come and push in and take the honey, take the wax, eat the brood. And in CCD, nothing was touching those colonies. So it was a very strange mix of symptoms where you have the queen is still there. It looks like they were booming just last week. There's not a pile of dead bees to indicate that they were killed off by something suddenly. They're just gone, and nobody else wants to steal the resources that are in there.
Let's begin the discussion with Ken Hashimoto on what has been learned in Japan about CCD.
I would like to talk about colony collapse disease, CCD, and neonicotinoid insecticide. CCD is a phenomenon in which worker bees from a beehive abruptly disappear, and neonicotinoid developed and have been used worldwide because they are believed to have reduced toxicity compared to previously used organophosphate insecticide. But neonicotinoid are suspected culprit of CCD. Firstly, I will review how European Union government deals this suspected culprit and how the US government does. In the case of EU, according to precautionary principles, EU has decided 2-year suspension of use for 3 neonicotinoids, which are thiomethoxam, imidacloprid, clothianidin. In the case of the United States government comments that according to on EPA and USDA's knowledge, none of the incidents that led to suspension have been associated with CCD. The CCD is a complex problem caused by a combination of stressors such as Varroa mites, poor genetic variations, malnutrition, and pesticides. And also, they comment additional research is needed to determine risks presented by pesticides. Now, I review the study of Dr. Liu at Harvard University. Dr. Liu proposes that true culprit of CCD is neonicotinoids. He said in Bulletin of Technology, volume 25 in 2012, that none of these potential culprits, except neonicotinoids, either alone or in combination, has been demonstrated to trigger the symptoms of CCD. And about the abrupt emergence of CCD in the United States during 2006 to 2007, he said that since most of suspected causes for CCD were not new to apiculture, there must have been an additional new stressor introduced to honeybee hives. And Dr. Lu proposed his hypothesis. The first occurrence of CCD in 2006 and 2007 resulted from the presence of imidacloprid in high fructose corn syrup fed to honeybees as an alternative to sucrose-based foods. And he rationalized as follows. Although the replacement of honey sucrose-based feds with HFCS among commercial beekeepers took place much earlier than 2006 to 2007, it was the timing of the introduction of neonicotinoids to the corn seed treatment program, first occurring in 2004 to 2005, that coincides with CCD. In order to prove this hypothesis, he gave high fructose corn syrup with neonicotinoids to honeybees as an alternative to sucrose-based food. And he demonstrated that high fructose corn syrup with a neonicotinoid causes the CCD. But his experiment is criticized by many researchers. The main point of the criticism is he carried out experiment by using neonicotinoids at unrealistically high dosage. Dr. Lu used 10 times higher dosage than the measured dosage in the field. Dr. Toshiro Yamada, he is a researcher, Japanese researcher, and also a beekeeper, which is very important. Dr. Lu is a researcher, but he is not a beekeeper. Dr. Toshiro Yamada measured photostability of neonicotinoids, and he found imidacloprid, which Dr. Lu used, is photoinstable, photolarval. Under sunlight, it decays quickly. It explains why Dr. Lu needs 10 times higher dosage than 10 times higher than measured field dosage. Dr. Yamada has done experiments in the field at a field realistic dosage by using a photostable neonicotinoid, dinotefran. Bees drink a lot of water in field. Dr. Yamada believes that neonicotinoid contaminated water in field causes CCD, but he did his experiment vice versa. He carried out his experiment in a neonicotinoid-free field. Instead, he fed neonicotinoid added sucrose solution at the realistic field dosage in the hive, so that he was able to measure exact consumption of a neonicotinoid, dinotefran. He clearly demonstrated that dinotefran causes CCD in 3 separate experiments. Lastly, I summarize the difference between Dr. Lu's study and Dr. Yamada's study. Big difference between 2 studies are dosage, photostability, which I already mentioned. As I mentioned before, Dr. Yamada is a beekeeper, a good beekeeper, so he didn't use apistan as a miticide and fumagillin as an anti-nosema drug, but Dr. Lu did. I didn't show, but Dr. Yamada demonstrated that CCD was caused by dinotefran, photostable neonicotinoid, but organophosphate, which is thought to be less toxic neonicotinoid, didn't cause CCD. Which is thought to be more toxic than neonicotinoids didn't cause CCD. Toshiro Yamada is a typical Japanese researcher who is a poor English speaker and a poor writer in English. So he published his study in a Japanese journal. That is why his study has not drawn much attention from Western society. I came from Japan all the way from Japan to present this his study. Thank you for your listening.
Are there any comments or thoughts on this short presentation?
Interesting. I think it's going to be interesting when see what happens in the next 2 years, or whenever the EU bans the use of those 3 pesticides. We'll get some interesting data to see if it makes a difference or not.
Are there other substances that are similar that have not been banned that could interfere with getting the kind of data that might be helpful?
I'm really not an expert on pesticides, so there's so many.
But are you familiar with any of the research that he was talking about, Bob?
The Harvard study, not this Japanese one.
What is your impression of the Harvard study?
Well, as you said, it got a lot of criticism because it wasn't a very natural situation. Mm-hmm. I think that just speaks to the larger issue of CCD, which probably we speak too much about in this country because we know that a lot of things kill bees, irrespective of CCD. I think all of us would argue that insecticides in beehives are a bad thing. Mm-hmm. Whether or not we ought to be banning these particular chemicals is certainly debatable, but we would probably be more productive talking about the things we know that are problems, like the mites.
Maybe you can explain a little bit more about that. I know you can.
Well, I'll just take my experiences, for example. I have never personally seen a case of CCD, and there has not been a case of this reported in my state of Louisiana. However, I can show you lots of colonies which are under stress, if not killed outright, by high infestations of Varroa mites. And several recent studies from several places in the world show that to be the major problem facing bees. Again, as pointed out in the presentation correctly, there are lots of problems impacting bee health. But I'm here because we do work against these mites, which are known to be a major piece of the puzzle. So.
No, I would say the same thing. I would agree that, you know, people talk about neonicotinoids, pesticides, and everything, but I don't think anybody can really point to one certain thing that this is the culprit. You know, so sticking to what we do know, just, you know, the things that we know are putting pressure on bees.
I've heard beekeepers say, I know what my CCD is, do you know what yours is?
Right.
So they all have a feeling, at least, of what's harming their bees most. I don't know how correct those impressions are, but there is quite a wide range of things out there. I spoke at a large beekeeping meeting not too long ago and said Varroa mites are known to be the big problem, knowing I was going out on a limb, and immediately had several beekeepers, no, it's the insecticides, and they might be right in their case.
Yeah, in my case, it's small hive beetle. You know, I mean,
have you had CCD in your hives?
I've never seen anything like that. I have seen pressure from the beetle, you know, wipe out a colony, but not anything that I would call CCD. You know, so I guess from where you live in, you know, in the United States is kind of probably determine. You know, I've heard beekeepers say winter is my culprit. You know, it's, you know, I can't get my bees to overwinter good, so that's a major problem, you know, with some beekeepers.
Not here, though.
Not here, though.
Well,
yeah.
I think also there's been some confusion about CCD because it was a strange phenomena that happened all at one time, and they characterized it with a few symptoms. And just because a colony collapses and dies doesn't mean that it's CCD. So when we talk about losing bees, most people in the US are not talking about colony collapse disorder anymore because that, like Bob, I've never seen it myself. It was a strange phenomenon and we haven't been able to recreate it. But there are mysterious bee losses that we haven't yet explained, and you do see the colony collapse. So there's some misunderstanding about that as well.
As you pointed out, the definition started one way, but it's been refined through time, which has been a good thing. Interestingly enough, there was a report just, I guess, about a week ago from the EPA and USDA which talked about colony mortality during the last year. And one of the reports that I heard about that report specifically pointed out that the incidences of CCD continue to go down, as we've kind of defined it down to something that's more reflective of the situation, that we don't really know what's going on. It seemed like a lot of people at the beginning were jumping on the CCD bandwagon. My colony died, I don't know what killed it, therefore it's CCD. That seems to be happening less frequently.
Yeah, I've been keeping bees for about 40 years, and in those 40 years there's been at least half a dozen major pests or problems that have come along, and these all sort of accumulate. There was chalk brood in the 60s, and tracheal mites came in the 80s on the mainland. Then Varroa mites came along the 90s, 80s, 90s. Yeah. And along with the Varroa mites became an effective vector of several viruses that had always been in the colonies but never had some blood-sucking animal that would transmit it from one bee to another. So we have all these viruses which are proving to be very toxic to the bees, killing a lot of bees. And then another Nosema serena, which is another major disease that's killing the bees, along with the new class of pesticides, which is still questionable whether it's a huge factor or not. All pesticides, or a lot of pesticides, are extremely damaging to the bees. They've been damaging bees for decades, and whether neonicotinoids are more or less damaging, I think we're going to be finding out soon with research that people are doing around the country.
Steiner discussed, how human society would be putting stress on bees, just the kind of life that we have and the kind of way we treat the colonies. And he predicted that there would be problems going forward in our society, and many people look to that as a way to point to human interaction causing stress. That is there anything that we can say about that relative to strengthening? When we had lunch today, we discussed, strengthening the hives, and what is your thoughts on that? On how to best strengthen beehives?
Well, there's no doubt that we've been our own worst enemy, human beings. Basically, it's the globalization where we're bringing things from one place in the world to another is what brings the pests, and of course we've industrialized the bees a lot, which stresses them, moving them across the entire country to go pollinate the almonds in California. That can't be healthy for the bees to be travel that far. But people have been moving bees for many years, and that isn't necessarily such a bad thing. But we haven't kind of industrialized things probably a little bit too much on the bee front, I would think.
Well, when we have such high levels of collapse, 30% or even 20% would be considered high in any industry, wouldn't you think?
Yeah, well, one thing you read in the papers all the time that 30% of the colonies die over the winter, and to a person that's not a beekeeper, you say, well, hey, in 3 years all the bees are gone. But what people need to realize is that bees grow in the springtime. Most bees die off over the winter. That's the most stressful time for bees, at least on the mainland. And when springtime comes, the ones that survive can be multiplied, just like you would multiply plants. So even though 30% of the bees die over the certain winter, usually by fall, those numbers have been made up, if not increased. And so even though beekeepers don't like to lose 30% of their colonies, so we've been sustaining that condition for a number of years. It's well over 10 or 15 years, I think. Ever since the mites arrived, that's when we've been having steady losses. But it's not so critical as non-beekeepers think. To lose 30% doesn't mean in 3 years all the bees are gone. you'd have to lose a lot more every year for that to happen.
Well,
I
thank you
think
for
also
explaining that.
it's important time and opportunity to point out what many people don't know is that the queen bees that are produced in Hawaii are critical for that spring rebuilding on the mainland in the US and in Canada. Because we can produce those queens early in the season here, many people are waiting for them in North America to rebuild from their losses. So the queen rearing industry is really important in Hawaii, not just for Hawaii, but for all of North American agriculture. There's also a lot of queens produced in California, more queens than Hawaii produces, but there are many more queen producers as well. And we have the advantage of being able to rear queens year-round.
You know, when these numbers come out with people reporting losses, if you guys, you know, talk with other beekeepers, would you say it was the backyard hobbyist beekeeper that these reports are largely based on, or the commercial beekeepers? There doesn't seem to be a distinction when you read about it in the paper of, you know, one or the other. You know, one of them obviously makes a living at, you know, keeping bees and is probably better suited trying to get his colonies to overwinter better, and the other one, backyard beekeeper who may be a part of a beekeepers association, reporting that, you know, I lost my colony, or half of them.
They do break up the data, so as I'm part of the Apiary Inspectors of America that started that survey, and now it's become a much bigger thing. But when you participate to say what your losses were, you say how many colonies you had. And in fact, most of the time the hobbyists lose more than the commercial guys on a percentage basis. But if you're a hobbyist and you lose 1 out of 2, you lost 50%.
Right.
So commercial guys are usually losing less than the hobbyists, which makes sense because the commercial guys are managing for a bottom line.
Right, right. No.
And the reason that colony collapse disorder became such a fast phenomenon was one of these really big beekeepers that had been doing everything the same for a long time and had generations of beekeeping in his family, all of a sudden lost almost everything.
Really?
So, and people went and looked at it and said, we don't know what's wrong, and we still haven't figured out what happened in that case. It's just a composite of symptoms that we haven't been able to recreate.
What would you like to see happen here in Hawaii relative to strengthening bee colonies?
Well, I think Hawaii has a great opportunity. I think it's very unfortunate that these problems that have been on the mainland for so long got here because we were protected by so much distance. But in the wake of all of that frustration, I think it's leaving behind stronger bees and better beekeepers. I think it'll make better management. So it used to be really easy to keep bees alive in Hawaii without really knowing how to manage them, without paying attention to their state of health. And now there are consequences if you leave your bees alone too long. If you don't manage them, they will perish. So I think that the beekeepers that will get through this will be much better for it. Although it's definitely a devastating loss to have your colony wiped out by beetle.
Are there any points relative to management that you might be able to suggest that beekeepers should, or wannabe beekeepers should look at?
Yeah, I think educate yourself is the most important thing you do. Getting into managing bees as livestock, it's really important that you know what warnings to watch for and know what you're doing. You can learn it the hard way, but you're going to lose some colonies. It's just like if you're going to keep chickens. If you just shoot from the hip, you're going to have higher losses than if you get some education and know what to watch for. So I think you can be much better at it. And in fact, you have to be if you want to keep your bees alive with these problems. It's really important to not move things around. We don't want anything moved. Right now, the Varroa mite, which is undoubtedly part of the whole bee loss story in the US and colony collapse, is only on Oahu and Big Island. So Maui has a great advantage that there's not Varroa mite here. But all it would take is one person to move something from one of those islands, and then that would be the next big crisis for beekeepers here. So I'd say educate yourself, don't take risks, get to know the beekeepers in your area and learn from each other. There hasn't been a strong network of Hawaiian beekeepers until now that we have this crisis. They're starting to talk and compare management
practices.
Yeah, and I think it's strengthening the community, and it's also making people like yourself aware of how important bees are. So,
Thank you. Bob, do you have anything that you'd like to add to that, perhaps?
No, Danielle did a fine job. You know, there is a lot of information out there. I completely agree with the notion of finding a local beekeeping group or mentor, because no one can do it like the people who live where you do can. And to me, the one bright thing coming out of the CCD situation is the amount of information we're gaining about how bees work and what keeps them healthy. So that'll be a good thing for the future.
Glenn had mentioned the commercial practices, and we talked here about the moving of bees relative to crop production and so forth. In the moving of bees, transportation of bees and preparing bees for pollination, there's a need to feed the bees. And we mentioned high sucrose feed for the bees. Are there any, in your experience, are there any other types of food for bees that might be helpful for strengthening bees? Any particular type of sucrose, perhaps, or a particular type of feed?
Nectar, plant flowers. I think in Hawaii, there's not a lot of people that need to feed bees. It depends on where you keep them and the plants that are available, but most of the beekeepers that I know in Hawaii aren't feeding their bees. They're just putting them in good, healthy forage sites, and there's a lot of that available in Hawaii. Bees are very productive here.
And in California, where the almond crops, of course, are attracting these huge movements of colonies to, on an annual basis, to pollinate, are there any other thoughts relative to?
Well, it's a completely different situation in California. In California, it doesn't rain for 6 months out of the year, and things dry up. And so feeding bees is necessary. And people usually feed either pure sucrose, white sugar, which as people we think is bad for us, but for bees that's actually their natural diet, similar to what's in the nectar of flowers. Or they feed them high fructose corn syrup, which may or not be good for bees.
Oh, I see. There might be a difference in the way bees react to the sucrose sugar versus the corn syrup.
It's thought that sucrose is better than the corn syrup, but there's research going on about the different effects. I know I fed high fructose corn syrup many years ago to my bees, and they seemed to do just fine back in the 1980s before we had other problems. But in recent years, I've only fed sucrose if I had to feed them.
And what was the reason for that change in your mind, in your thinking? Was it to try to eliminate possible factors that you were not sure of?
There's been some questions about high fructose corn syrup in bees. It's really not that unnatural for bees. Well, high fructose corn syrup is fructose, and that's a sugar that bees.
Has there been research done relative to corn syrup that has indicated there might be a difference between the cane sugar and the fructose?
There has been research. I know if the corn syrup, high fructose corn syrup, is old and been heated, there's a certain toxin that forms that's not good for bees. So very old corn syrup has been shown to be bad for bees. I think people have always wondered about corn syrup being produced from BT corn.
BT is, it's a chemical substance, I believe.
Well, it's a naturally occurring substance from, what is it? It's a bacteria.
Bacillus thuringiensis.
Yeah, Bacillus. And, but as far as I know, the research hasn't shown that to be harmful to bees.
People are talking about the mutation of the Varroa mite. I'm not very familiar with it because we don't have it here. Are there any works you guys do that somehow we're creating a stronger, more resistant mite?
Oh, I see.
Yeah.
Yes. Mites as a model organism are like the poster child for developing resistance to chemicals. And that has happened with Varroa for some of the first synthetic miticides that we've used against them. So some of these chemicals certainly have lost a lot of their efficacy, don't kill mites as well as they used to. Beekeepers get caught off guard by this, and again, the mites become a problem.
And then so with the VSH strain, the Varroa sensitive hygienic, Is there any work that needs to be done to keep up with this mite? If, you know, if it's getting more resistance to you know, chemicals, can it become more resistant to the bees' actions?
Probably not.
Probably not.
We'd have to go into a description of what goes on with this behavior, but
Right.
hard to imagine.
Hard to imagine.
Yeah.
Well, I think the main thing is with a chemical, so if a Varroa mite can break down fluvalinate, which is the single chemical that we would put in Apis strips, and you put it in there and it kills 999 mites and one survives, that mite has an enzyme that can crack it, so it doesn't kill it. So it's not that that mite is a super mite and can take on any chemical, it's just that that one's not going to kill it. So you lose the tool from your toolbox. No longer can you use that to kill the mites.
I see.
And with the VSH-B, it's a behavioral mechanism.
Right.
They're sensing it, they uncap it because they know there's a Varroa reproducing in there, and that is a lot less of a lock and key that can be cracked.
Right.
You know, if you've got one chemical and the immune system can break it, then that mite will just proliferate in your hive. So VSH is a much more robust mechanism to manage mites than a chemical.
Yeah, we haven't really described what VSH is. That's
Please do.
actually that's why I'm on Maui right now, is to help Paul actually breed bees that are naturally resistant to the Varroa mite. Now, fortunately, the Varroa mite is not on Maui, but the queens that a person produces on Maui and exports somewhere else would be good if they were resistant to Varroa mites. And if Varroa mites ever get to Maui, it'd be really nice to have the bees already resistant. As well as there's a couple other diseases that the VSH bees are resistant to already, American foul brood and chalk brood. So anyway, Bob,
Those are bee diseases.
those are other serious bee diseases. Bob actually is the expert on VSH because their laboratory developed the naturally occurring trait. Why don't you describe what it is?
The basic concept here is to try to make bees which are genetically disposed to eliminate the mites on their own, so we don't have to put chemicals in the hives. And these bees, which scientists at the lab discovered and bred, increased the level of this behavior, target mites that are reproducing in the hive and at least interrupt that reproduction, which eventually flattens out the growth rate of the population of mites in there. It's not perfect, but it works pretty well, and in most circumstances these bees can keep the mites below damaging levels. I will say up front that it's going to be more difficult in Hawaii. There's a lot of mite pressure here, so it probably takes a greater level of this behavior being expressed by bees than it would be in other places where the mite pressure isn't so great. But there's no question that, again, to use your term, it's another tool in the toolbox which holds good promise.
When we talk about selective breeding, of course, as scientists, it's understood that this is kind of what happens in nature and how evolution occurs. Now, of course, with the genetic code being broken and so much lab work being done on modifying animal behavior relative to genetic engineering, there have been discussions that relative to genetic engineering of bees. We in our society see, so many problems, unintended consequences of human actions, that we do something because we can, not necessarily because we should. What is your opinion of a genetically modified bee? For me, it's abhorrent to think of because of the unintended consequences that we find in so many other areas of science and of our lives. But you as experts, what would your thoughts be relative to that kind of scientific play? I
don't know anyone who's talking about genetically modified bees in the sense that you're using that term. But as a theoretical construct, what would you have to tell those companies or those people that are involved in genetic modification? What would your guidance be to them? Well, again, we're not anywhere. It's so far from reality that we're not even thinking about it. I understand what you said, that there are unintended consequences, but I do need you to understand that that's not what we're talking about here in this particular case.
Now, the VSH trait was a trait that the bees had in their collective genome for, for millions of years, probably somewhere in the past. There was a mite, maybe it was Varroa, maybe it was something similar to Varroa, that they had this resistant trait to get rid of it. And then when that pressure from that pest went away, the genes that control that trait just kind of laid low in the genome until now when we were able to bring it out through selective breeding. So this trait is a perfectly natural trait. That what we're doing as beekeepers, is kind of working with nature, working with what the bees have already developed, and just expressing it more quickly than what happened in nature. It might take hundreds of years to get to a level where it's going to would be useful, but now we're bringing it out so beekeepers can use it now. But personally, I started using the VSH bees soon after it was developed at the USDA lab, and I was able to stop using any chemicals in my colonies at that point, and for the last 12 or 13 years haven't had to use any chemicals for Varroa mite control at all. So,
well, that says a great deal.
I know it works. Yeah.
That says a great deal.
And that's why I'm here trying to help Hawaii bees become resistant on the Big Island, where they already have Varroa, and on Maui, where we can get ahead of the curve for a change. Maui is one of the last places on Earth, Maui and Kauai, last places on Earth that don't have the Varroa mite. It's only other place is Australia. So it's a unique opportunity to get ahead of the game here.
Well, and to learn to do things with smarter tools than just the next best chemical.
Exactly. I got on that chemical treadmill when the Varroa mites first hit us in California many years ago, and I did what they told us we had to do. Well, I saw the losses, first of all, when the Varroa mites came, and they were severe losses when the bees had no protection against Varroa at that time. And so we started using the fairly benign chemicals, the apistan strip. But it wasn't too many years before, as Danielle said, the mites became resistant to the chemical we were using. Then they gave us another chemical, Coumaphos, which is organophosphate, which scared me to death because I knew that's toxic, very toxic to people. And it was shortly after that, fortunately, the USDA came up with the VSHBs. And I remember very clearly that I was using this Coumaphos. I didn't want to. And I was working my hives out there, and I was, sometimes you're working and there's a nice big chunk of comb honey that you pop into your mouth, and it's warm and delicious and wonderful. And I was just about to pop that in my mouth, and I saw this chemical strip in my colony, and I said, oh my God, I don't even want to eat my own honey here. This is ridiculous. And fortunately, the resistant bees came along just about that time, and I said no more, do or die. And fortunately, the resistant bees saved my beekeeping career.
The chemical use thing just kind of scares me. I hear about the guys on the Big Island, and it's like, wow. Some of these things are killing the mite, and I don't know how they are now, but at one point I was hearing they're almost killing the bee too. It's like you got two insects living in a box, you're trying to kill one and let the other one thrive. It's a tricky situation. So, I'm with you. I'd rather get this, and that's why we're doing this. Rather get these bees up and running over here, so that if anything does happen, maybe won't have to go that route.
That's what we all hope.
Yeah.
Is there another type of strain of bees that are being looked at as far as particular habits that might be good to grow, or are we kind of at the end of that in our understanding at this point?
No, there are people around the world doing work on this topic. It's it's an obvious, correct way to do things. But it's been a very, very tough row to hoe. I mean, the Europeans worked on this for decades without really getting anywhere. There are some potential traits out there that have mixed results so far. But as we talked about this morning, there are really 3 types of bees with documented resistance to Varroa. This VSH type bee. We have another bee at the lab that we imported from the far east of Russia, which evolved in an area, or at least the longest, they've had the longest exposure to these mites, and they have some resistance. And then there's a more generalized form of hygiene, the H in VSH, that was developed in bees at the University of Minnesota, which also have some resistance to Varroa. Those are documented to exist. There are lots of reports out there about surviving bees which are resistant, which may be true. There are bees reared by commercial beekeepers who claim their bees are resistant, which may be true. They're just not documented. But I can assure you that there's plenty of research going on into enhancing the range of possibilities for resistance.
In the beekeeping communities worldwide, is there a reluctance to share information, or is there an openness to share information? What's the culture like?
Very open.
Very open.
Very open.
With the internet, I suppose that makes it this information even more accessible.
Well, to me, as a scientist, it's encouraging. It's often the case these days where research groups get grant money, which is what keeps a lot of people going. It is written into the grant that the data produced must be what they call open access. It has to be freely published somewhere where everyone can see it.
Well, I want to thank you all for coming. Everyone loves honey, and no one wants to kill honeybees. And perhaps we'll meet again and talk some more as we learn more about bees and best agricultural purposes. Thank you for sharing. Thank you for coming.
Thank you, Larry.
Thank you.